advanced biomedical scientist. I work in microbiology department in the pathology lab at the Royal Oldham Hospital. My main job is a supervisor role, but what biomedical scientists are very much involved in is they analyse samples that come from patients who are in hospital or patients who have been to see their GPs. The microbiology biomedical scientists will analyse those samples, looking for causes of infectious diseases, mainly bacteria and viruses, but we can also look for other things like parasites and fungal infections. Our main focus since March has been a little virus called SARS-CoV-2. You may also know it as coronavirus or COVID-19. Currently, the department are processing around 1,000 swabs a day for coronavirus, mainly from our hospital patients. We started looking at these in March, and we've had to massively upscale our COVID testing since then, as you can imagine, because it was only around about a year ago where the first, in fact, it might have been a year ago today, where the very first case of coronavirus might have actually been recognised, although we didn't know about it yet. As well as COVID-19, we also look for bacteria you may have heard of, such as Salmonella, Shigella, E. coli, which are common causes of food poisoning. We look for the bacteria that cause things like septicemia and meningitis, such as Neisseria meningitidis, Streptococcus pneumoniae, and Haemophilus. And we're also very much involved in looking for superbugs such as MRSA and other antibiotic resistant bacteria. We huge role in informing the doctors and nurses what antibiotics they can use to treat a patient. So once we've isolated a bacteria, we will test different antibiotics to see which one is the most appropriate for the patient, given the symptoms and given what infection they might actually be suffering from. We use various different techniques for finding these bacteria. I mean, obviously, I'm sure you all know bacteria and viruses are invisible to the naked eye. So we use microscopy techniques for things like parasites. A lot of the microbiology techniques that look for bacteria are quite manual. So we still do something called culture, where we will take a patient sample, plate it out onto an agar plate, which is like it's a jelly plate. It's solid at room temperature. It's got all the nutrients in that bacteria needs to metabolize, and then you end up, after 24 hours incubation at 37 degrees, you end up with little tiny colonies on a plate, and these are the things that you can manipulate to identify the bacteria and to find out what antibiotics it's resistant to. For viruses in particular, we can't use those sort of techniques. Virus detection is very much the more modern area of microbiology, and you may have already heard about all of these in the media, because obviously the big focus is on COVID-19, but we use in the main a procedure called PCR, polymerase chain reaction, and what that does is it takes a small segment of DNA or RNA which is specific to that virus. It uses enzymes and amino acids to amplify that virus to a detectable level, and then the various analyzers we have in the department will be able to tell you what virus it is based on the section of DNA or RNA that it has detected. Again, viruses that you may have heard of that we can detect in the department: COVID-19, hepatitis, HIV, things that cause chickenpox like varicella, and then various other viruses that might cause diarrhea and vomiting, norovirus, rotavirus, and then other viruses that may cause skin infections like herpes, varicella, and there's so many different viruses. I think there's hundreds more viruses than there are bacteria, and there are probably lots of viruses out there that we aren't even aware of yet. As well as using PCR to detect viruses, we can also look for antibodies in our blood. So the viruses that we still haven't found a genetic code for, we can use a technique called serology, and that is actually detecting the antibody that the human body has raised against the virus. HIV diagnosis is usually made based on an antibody detection rather than detection of the virus itself. So if you suspect that you may have had contact with a person with HIV, you can get a blood sample done at your GP, and the GP will ask for the antibodies to HIV rather than HIV itself. Same goes for hepatitis as well. If a patient is found to be hepatitis positive or HIV positive, then we can take a sample of the blood to look for the virus in the blood. And again, that technique that we use is PCR. Other branches of microbiology, such as fungal infection, we can diagnose things like athlete's foot, and things like, the name just escaped me then for a minute, skin infections and scalp infections. Generally, a doctor will take a sample of skin scrapings or a nail clipping and send that to us. This, at the moment, we're using a very traditional technique for fungal infection. It can take 3 to 4 weeks to grow a fungus up, and one of the things we want to do when we've got ahead of the COVID pandemic is to actually use a PCR method for detecting fungal infections. The focus for us is to try and get results out quicker than 3 to 4 weeks, so that's something we want to look at in the future. Parasites that we look for as well, we mainly look for parasites that may cause diarrhea and vomiting, and a lot of these are usually acquired abroad on foreign holidays. One of the most common parasites in this country is one called Cryptosporidium. Parasites, we use microscopy to look for those. Cryptosporidium is a really nice one. We use a fluorescent stain. We take a sample of poo, essentially, smear it onto a slide, and then use a special stain called auramine. We look at that in a dark room under a microscope, and the Cryptosporidium parasite, you can just see the eggs in there, and like little green circles that fluoresce really brightly on the microscope. So that's one of my favourite ones to look for. We also look for a genital pathogen called Trichomonas, mainly affects women, and we will take genital swabs, do what we call a wet film, and we can look at that under the microscope as well. And we usually see live Trichomonas as well. Other things that we tend to do in the lab, we do a lot of quality assurance. Obviously, we've got to make sure that we're reporting the right result on the right patient at all times. So we do a lot of quality control checking. So we will run known positive samples through our analyzers to make sure our analyzers can pick them up. One of the things I've been heavily involved with in the last few weeks or few months is verification of new equipment. We've had a lot of new assays and platforms introduced into the laboratory to detect COVID-19. Given that we didn't know about this virus a year ago, the pharmaceutical companies, the scientific companies have very quickly had to step up and create these assays. A lot of these assays usually take 2 or 3 years to come to market, whereas some of these were available in 2 or 3 months. So we have to make absolutely sure that they are fit for purpose. So my main role over the last 6 to 9 months has been bringing these assays into the lab, testing them with samples that we know are positive because they've been identified elsewhere by known methods, running them through the analyzers and just making sure that all the results are as they say they are. A lot of manufacturers will publish claims such as how sensitive the assay is. So if you've got 100 positive samples, will it pick up all of those positive samples? That's sensitivity. We've got to prove that that analyzer matches those sensitivity claims. What else do I do in the lab? A lot of my job now as well, I'm a supervisor, so I do a lot of HR. I get involved in writing staff rotas, planning shifts, making sure that we've got enough staff in the lab to cover all the shifts that we need. We've had quite a big change this year with our shifts. We used to work from 8 o'clock in the morning to 8 o'clock at night, and then 9 till 4 at the weekends. but the last few months, to cope with COVID, we've had to increase our working day till 10 o'clock at night, sometimes midnight. We've had to put on night shifts. Weekend shifts have significantly increased from 9 till 4 to 7 till 10 at the weekend, just to cope with COVID. I don't think there's much more I can tell you, really. That's a quick 10 minutes on my job. I don't know if anybody has any questions that they want to ask.
If anyone's got any questions, you can type in the chat box and we can read them out for you.
All right. So we've got a question from it. What course did you do in university?
All right. Okay. Well, first I'll start at college first. The very first thing that I did was I did A-levels in biology and chemistry. I must admit, when I was at college, this wasn't the career path that I'd considered, and I'm guessing, well, I was 16, 17 at the time, which is, I'm sure, is the same age as the people listening. I hadn't heard of this job. At the time, I wanted to be a vet, so I did biology and chemistry. During my biology A-level, when you get to know a little bit more about the human body and infectious diseases and stuff, I realised I was interested in microbiology. I went to the University of Birmingham. I did biological sciences with a specialism in microbiology. And it was only at uni, when I was at the in our careers office, where I actually found out about being a biomedical scientist. The route that I took is probably not the most direct route. And bear in mind, I was at university 20 years ago, a long time ago. Things have moved on since then. So when I left uni and decided to become a biomedical scientist, I actually had to do a top-up degree. My degree was microbiology, but it was very broad, and there was a lot of modules in there about plant biology, genetics. So some of that, although was very worthwhile learning, and it has put me in good stead for the job, it wasn't a prerequisite for the job I'm doing now. So I ended up going to Manchester Met and doing a top-up degree in biomedical science. I think that is the way I would advise people to go now. If you were interested in being a biomedical scientist, I would definitely look at the biomedical science degrees. I know Manchester Met offer them. I think we've got a couple of lads here who have just started doing biomedical science apprenticeships. I think they're going to Macclesfield for those. What I would suggest as well, I'm a member of a professional body called the Institute of Biomedical Science. They have a really good list of accredited degrees that are relevant to biomedical science. So if it's a route that you know you want to go down, just Google Institute of Biomedical Science. It's a really good website. There's lots of different tabs on there for career progression, how to get into the job. And like I said, there's a list of accredited degrees. I know there are a couple of universities in the northwest and further afield that do accredited degrees. Some of the accredited degrees as well, they give an option of taking a year out and working in a lab. And we do have a few students here who, I think the course is a 3-year course. They start off sometimes doing like a day release in the lab, and then when they move into the second year, they spend 2 or 3 days here. And then the third year is a full year of working in the lab. To become a biomedical scientist, you also have to be state registered. So you have to complete a specialist portfolio that is through the Institute of Biomedical Science. And some of these degrees allow you to do that portfolio while you're doing your degree. So by the time you graduate from that degree, your portfolio is signed and you're good to go. You're good to get a job. We have employed quite a few students who have gone down that route. There isn't always a job guaranteed at the end, but if you can get a placement in a lab, it's a really good foot in the door. Thank you very much for that one.
You might have already answered this, but Hannah's asked, how long was the course?
Yeah, my course was 3 years. What I didn't mention actually was the top-up degree that I started off as a postgraduate certificate, but then I carried on for another 2 years and ended up doing a master's. My position as an advanced biomedical scientist requires a master's degree. I did the Masters part-time, so that added on another 3 years. I think the first 2 years were part-time, and then the third year was a project course, which I managed to do, as well as working a full-time job. The biomedical science degrees, yeah, are mainly 3 years. Some of them are 4. It all depends on which route you want to take.
Thanks. And Kenzie's asked, do you come across any job?
Not in my job particularly, but we do have biomedical scientists who work in biochemistry. That's mainly analysis of patient samples for things like hormones, various biomarkers within the body that indicate liver function, kidney function. That is more, yeah. So the chemistry part is more of a biological chemistry side. But I mean, I did a chemistry A level as well, and that has given me good skills in things like what do I need to do in my day-to-day work? Things like making dilutions, making up acids and different reagents. So there is a little bit of chemistry in there, but like I said, the biomedical scientists have a bit more of a biomedical chemistry element going on in their job.
Brilliant. And Afsa, do apologise if I do pronounce anybody's names incorrectly, has asked, what do you do on a typical day in your job?
All right, okay, typical day in my job. I come into work, I usually open up my emails first, catch up on my emails, get emails from all sorts of people, service users, managers, our consultants. Some are requests to chase up tests that are outstanding. Some of them are asking for input on various standard procedures and things like that. At about 9:30, then we'll have a senior huddle. So myself and the other advanced biomedical scientists will just sit down for 10-15 minutes. We'll have a chat about if there's any staff absence that we need to cover, if we've got any engineers coming in, workload, do we need to manage the workload a little bit differently today? Following that, I'll usually have a walk around the department, see how everybody's doing. And then after that, for me, I review. At the moment, I'm doing a lot of reviews. I review a lot of our standard operating procedures. So that's the guidance that we use to make sure we're doing the job correctly. I'm involved in audits as well. I don't think there's a typical day in biomedical science, to be honest. The biomedical scientists who are on the benches and doing the bench work, they'll usually start off by, they'll get all the culture plates out from the incubator from the day before. They'll sit and read. When I say read, what they're doing is they're examining the culture plates for different types of bacteria. They'll spend the morning doing various tests to identify what that bacteria is. And then the afternoon is usually spent testing those for antibiotic resistance. We get urgent samples in as well, things like lumbar puncture fluids. So CSF, which stands for cerebrospinal fluid, they're urgent samples. They need to be looked at as and when they come in. We do a microscopy for those to look for different cells in there that are a good indicator of infection. We usually fit microscopy in there during the day. So like I said, the cryptosporidium parasites, somebody will go and spend half an hour looking at those slides. There are no typical days, to be honest. It all depends very much on what's going on. To be honest, it's difficult to sort of convey over a Microsoft Teams call what the biomedical scientists do, because a lot of it is very visual. You need to be in the lab and see what they're dealing with on a day-to-day basis when I talk about culture plates and microscopy and things like that. So I hope that's answered your question anyway.
Hi, has anyone else got any other questions they'd like to ask Cheryl?
Hi Cheryl, so I'm the employability coordinator at Rochdale Sixth Form. I'm just wondering, when COVID isn't around, is there any opportunities for students to do work experience in labs, and how would they go about getting a placement if they were able to?
Hi, so is it Gemma?
Yes, it is.
Yeah,
Hi.
yeah. Hi Gemma. How old are your students? 16, 17?
Yeah, 16 to 18.
16 to 18. Yeah, we don't, you should be okay. We usually do allow students in over the age of 16 into the lab. I think the best thing to do, I can get in touch with you outside of the Teams meeting if you like, with our training manager. He's the best person to contact with regards to work experience. It will be very much a visual thing. They won't be able to do any procedures, but we definitely, once the COVID pandemic is over, whenever that will be, we do welcome students to come and have a look round and see the lab for themselves, really. But our manager would be the best person to contact, and I can give you those details outside of the Teams call.
That would be great, thank you. I've just had a student message me asking what the typical salary for a biomedical scientist looks like.
All right, okay. When you graduate from university or whichever route you've taken, you will go into a trainee post that starts at there's a banding system across the NHS called Agenda for Change. A typical trainee biomedical science post is a band 5. Forgive me because I don't have it in front of me, but it usually starts at about 23,000 a year. And then there's an incremental scale, so every year you'll get a pay rise, and I think the band 5 goes all the way up to about 30,000. In the lab, once you've completed at least a year of a trainee role and you've got your registration portfolio signed off, you will move up to a band 6, and that starts at about 27, 28, and that can go all the way up to 35, 36,000. I'm an advanced biomedical scientist, and again, that's on a band 7, so that's an increase on the band 6. There's usually opportunities to claim overtime and enhanced rates of pay on top of that for working night shifts, on call and weekends as well. But again, Agenda for Change is the thing to look at for more specific salary rates, I think.
Thank you. And we've just had another one of what other career paths can you do with a biomedical science degree?
Oh, good question. I probably should have mentioned actually that we don't just have biomedical scientists in the lab, and we do have other grades of staff. We have what are called an associate practitioners. They start on a band 4. They don't necessarily have a biomedical science degree, but a lot of our associate practitioners have started from scratch, really, and they do go to university and do a part-time degree as well. But some of them do have biomedical scientist degrees. So our associate practitioner, they will do other things like they can do everything except report and do anything that generates a result. So they'll run machines and analyzers. We also have biomedical support workers who will do a lot of the data entry and the specimen preparation. Some of those have got biomedical science degrees, but it's not necessary for that post. I'll be honest with you, I think having any sort of science degree is a good basis for any scientific career. I'll be honest, I don't think that there are any other careers that specifically require a biomedical science degree. And like I've said as well, I didn't come into this with a biomedical science degree. I've come into it with a biological sciences degree. Something I wanted to get into was forensics. Biomedical science degree may be a good grounding for that. It won't be the exact degree that you need, but it gives you some skills to become a scientist. I've already mentioned we have biochemist, biomedical scientists, but we also have people who work in histology. So they deal with cellular pathology and post-mortems. They work in the mortuary, so that's another career path you could go down. I can't think of anything others, to be honest. Like I said, I think just any science degree is a good foundation to get you into any science course. There's a lot of quality type roles out there in science. So things like food safety, food quality, pharmaceuticals. Biomedical science degree would give you a good grounding for a career like that as well. Okay, thank you.
So, and we've got a one from Assad. Is your job related to generic engineering by any chance?
No, it isn't. There's no engineering involved in my job, unfortunately.
I can't see any. Does anyone get any more questions? If you want to put it in the chat, and then I can read them out to Cheryl.
Okay, if there's no more questions, we'll wrap up for today. If you think of any questions that you'd like to ask after this meeting, you can email them to Gemma, your tutor, or to the work experience at srft.nhs.uk, and I'll forward them on to Cheryl, who will, I'm sure, gladly answer them. Thank you, Cheryl, once again for today. It's been very informative, and thank you everyone for attending.
Thank you very much.
Thank you. Thank you to everybody as well. Thanks for listening.
Thank you, Cheryl. It was brilliant. Thank you.
Thank you.